Do you know what keeps your metabolism going?
No, but I'd like to hear about what goes inside my body. After all, I should know where this burger ends up.
Well, this yummy burger that you're having gets broken down into carbohydrates and protein that are used to fuel your body. Your body then breaks down those carbohydrates into glucose, which is a form of sugar, and those glucose molecules then enter glycolysis.
What is glycolysis?
During glycolysis, one glucose molecule breaks into two molecules of pyruvate. The pyruvate form gets converted into a molecule named acetyl-CoA, which enters the Krebs cycle, another metabolic process that stores and transports our energy, ATP. ATP, which is a form of energy, is made through the means of enzymes. And enzymes are proteins that speed up chemical reactions. A change from pyruvate to acetyl-CoA begins the Krebs cycle, and it all occurs through the molecular machine called pyruvate dehydrogenase complex. Located in the mitochondria, the pyruvate dehydrogenase is composed of three different subunits named E1, E2, and E3. Now let me get a little sciencey here. E1 and E2 are involved in the direct formation of acetyl-CoA, whereas E3 is a FAD-NAD plus dependent system, some molecules that aid in the process of turning pyruvate into acetyl-CoA. Details of the subunits are for some other time, but I mainly want to discuss the happenings of the E1 subunit. The first step is pyruvate binds to the E1 subunit of PDC, and a carboxyl group is removed from the pyruvate, forming a carbon dioxide.
I get it, and since you said there are two molecules of pyruvate produced per glucose molecule, there are two molecules of carbon dioxide produced.
You got it. So to produce two carbon dioxide, this process occurs twice. Next, a molecule called hydroxyethyl TPP moves to the E2 subunit of the PDC, where it is then attached to CoA, forming acetyl-CoA. This process is a little more complicated than that, but for our purposes, the important part is that acetyl-CoA gets formed. Do you see where acetyl-CoA would go now?
Oh yeah, we begin the Krebs cycle from here.
Absolutely. The acetyl-CoA enters the Krebs cycle, a major step in cellular respiration that provides us with oxygen at the end.
I'm wondering what happens if the molecular machine breaks down.
Well, the machine breaks down due to mutations, which are changes to the structure that lower the protein's activity. As PDC's activity lowers, pyruvate would no longer be converted to acetyl-CoA, causing it all to build up. Pyruvate buildup causes lactic acidosis, which is the conversion of pyruvate to lactic acid. And too much lactic acid in the body can be toxic.
Oh boy, so one mutation and we start losing our breath.
Yeah, and not only that, but failure of PDC to function properly also leads to diseases such as obesity, diabetes, and worst ones of all, cancer.
Cancer? How is PDC involved in cancer?
Cancer occurs due to unregulated cell growth. In other words, cancer cells just keep growing and multiplying, and their resistance to the body's normal systems is what makes people sick. When the PDC is inactivated, the body's normal metabolic processes are interrupted, and this enhances cancer cell growth.
Inactivated? Like by another mutation?
Possibly. It could be due to a mutation in the gene that expresses PDC or inactivation through a process called phosphorylation, which is when a phosphate from ATP is added to the PDC at the E1 subunit, carried by enzymes called kinases. Since the PDC is important for metabolism, it is highly regulated by kinases and phosphatases. The PDC can be reactivated by a process called dephosphorylation, which occurs when an enzyme called phosphatase removes the phosphate group from PDC that was previously attached by the kinase. So just because the PDC gets inactivated does not mean it can't be reactivated.
That's so interesting. I would love to learn more about molecular machines.